So today we'll look at the topic. The topic is morphology and physiology of bacteria. Okay. Now, why is it important? Because understanding the fundamentals of this will help us in understanding how various types of bacteria cause human disease, understanding the evolution of the disease, and the diagnosis of the same in the diagnostic microbiology lab. Okay. Now, let us start. William Osler said that fever is among the three great. Humanity has three great enemies. The biggest one is by far is fever. So long throughout the human history, fever was associated with disease. However, now look at this chart. This is our understanding of human diseases throughout the modern evolution. Initially, there were many diseases. They were called as plagues. Plagues means some diseases which used to affect huge number of people, leading to morbidity and mortality. Thereafter, as you can see, on the ascent of the 19th century, we were able to identify various organisms. Our understanding has changed drastically from implicating environmental factors as disease to specific causative organisms. Classically, if we look at the history of medicine, there is a traditional and classical history where mainly the effort was given for the diagnosis from the clinical symptoms. Now look over here in the early modern medicine in the 19th century, there was discovery of pathogenic bacteria, obviously with the aid of microscope and further evolution. Thereafter, in the late modern medicine, that is after 1900, there was discovery of antibiotics, and that led to a tremendous change in our understanding, and it also aided in better treatment and outcome regarding infectious disease, especially caused by bacteria. Having said that, look at these three pictures. The first picture is of Frederick Gustav. He was one of the pioneers about describing about human diseases before the discovery of pathogens, and he labeled the diseases as miasma, contagion, or miasmatic contagions. Thereafter, next comes Louis Pasteur. Now, one of the biggest contribution of him is disproving the theory of spontaneous generation. Now, what was this theory? This theory led to believe that diseases generated or it generated spontaneously within human beings. But he disproved them. Thereafter, Robert Koch, one of the very pioneers of modern microbiology, he first saw the bacteria under microscope, drew the picture, and thereby labeled them as bacteria. So this is the evolution of causative pathogen of bacterial disease. Subsequently, many diseases, as you can see in the list, and their implicating bacteria have been identified. Over the period of the last century, these diseases were further more elaboratively understood, and the treatment of these diseases, especially with antibiotics, really gave a very good effect on the human population itself. As of today, we have controlled many of the bacterial diseases. Many bacterial diseases has been prevented by vaccines. The most notable one is tetanus. I think most of you are all vaccinated against tetanus, and the cases of tetanus have reduced drastically, not only in India but worldwide. But having said that, on the 21st century, we have arrived at a new kind of a problem, and this problem is stated as antimicrobial resistance. That means the very antibiotics or the drugs which were used for treatment against bacterial diseases, now we are observing that most of these bacteria have become resistant to the antibiotics. Now, so to understand this problem as a whole, we need to understand what bacteria are, how they function normally, and what we can do so that we can, which leads to better diagnosis of bacterial disease and better treatment outcome. This begs to the question, what is a bacterium? Any idea? I think you do have some knowledge about prokaryote and eukaryote, right? So, what is a prokaryote and what is an eukaryote? This is a primitive cells, predominantly unicellular. They don't have a defined nucleus, as you have rightly said. So, the origin of the word comes from the Greek letter bacteria, or it means staff or cane. Now, why it was named? The first bacteria that was observed was Bacillus. And if you look at the picture, it looks like a cane or a stick, large stick. So, from there the word comes bacterium. Now, singular is bacterium and bacteria is a plural terminology. Okay. Now, look at this picture. This picture shows the size of various cells. Now, eukaryotes are the largest cells, whereas viruses are the smallest living beings, right? So, prokaryotes are in between the eukaryotes and the viral. And if we look at the classification of bacteria, there are many types of classification. The first one, if you look at this, I think you will be much more aware of this one. The living organisms have been classified into two parts, the plant and the animals. Those are the primitive classifications. Again, we can differentiate into Plantae, Protista and Animalia. Just try to recall what you have learned in biology in your +2. Nowadays, the classification has been described as this. We have the unicellular organisms as either bacteria or archaea, and rest are all eukaryotes. Okay. Now, if you look at the lineage, the unicellular prokaryotes have two lineages. One gives rise to the bacteria, the other is archaea, and among these bacteria, there are various types of genus and species. Now, this I am talking about all bacteria. Now, keep in mind, we are trying to learn about medically important bacteria, not all bacteria, right? So, there is a very important thing that you must understand, that is called as classification of bacteria. How we can classify? The previous classification that I had showed, that is also called as the systemic classification, that encompasses all bacteria. But when we talk about medically important bacteria, we don't need to go through all that classification. The most important classification is called as the phenetic classification. Now, what does phenetic classification means? It means when you try to observe the morphology of the bacteria and classify them according to them is called as phenetic classification. That, now look over it. What is based on? It based on cell shape, size, morphology, ultrastructure, that helps us in identify a particular bacteria as quickly as possible. See, in diagnostic, we need to identify as quickly as possible. Okay, so there, this phenetic classification is very important. Next comes is the chemotaxonomy. Now, chemo means obviously chemicals. So, based on the chemicals. So, what it actually means is arrangement of bacteria according to their chemical nature and most importantly, metabolic and physiological activities. Okay. So, what kind of materials they require for their normal physiology? What are the substances they take up? What are the substances they give out? What is the environment they grow in? Based on that, we call it as the chemotaxic classification. And finally, the most important is called as the molecular classification. Now, all bacteria will have a genetic material, that is the DNA, and those genetic material are different in various genus of bacteria. If we classify according to the differences of the genetic material, we call it as molecular classification. These are the three predominant type of classification. Now, why I am showing this? Because in diagnostics, the most important is phenetic classification. Now, for phenetic classification, the requirement is understanding of the various morphological pattern. Now, let us look at why this is important. Now, look at this chart. Medically important bacteria can be classified based on the shape. What are the shapes? I think you already know that bacteria has a cell wall, right? What is cell wall? It's a covering outside the cell membrane. Okay. Human cells don't have a cell wall, but the bacteria do have. And what is the function of the cell wall? It protects the bacteria from the environmental elements. But there are some bacteria which don't have a cell wall, or some have very rigid cell wall, and some may have very flexible cell wall. Again, among the rigid cell wall, what could be the shape? It could be simple unicellular or it could be filamentous. Filamentous means very long shape. And among the unicellular, it can be either an obligate intracellular or it could be free living. Now, what does it mean? Obligate means absolute. And what does intracellular means? Over here, intracellular means inside human cells. Okay, so that means there are some bacteria which cannot survive in the environment. It can only survive within the human cells. Those are called as obligate intracellular. Whereas some other bacteria, it can survive in the environment. Those are also called as free living. This is one type we can classify. Another type based on what is called as based on gram staining. I'll explain it in more detail when I'll be talking about the cell wall. Now, this is a staining method which differentiates cell into predominantly two types. One is called as gram positive bacteria, another is called gram negative bacteria. Now, this is a very important step. Why? Because cell wall is one of the particular areas which is acted upon by most of the antibiotics. If the antibiotics act on the cell wall, what will happen? Some antibiotics will be more effective in case of gram positive bacteria, and some will be more effective in case of gram negative bacteria. This helps us in choosing the antibiotics when we are defining the treatment regimen with antibiotics. Okay. Now, according to the morphology, how can we describe? These are very simple terms. This is called as coccus. Coccus means it's a circular cell. This may be aligned with two cocci side by side. If that happens, we call it as diplococci. Diplo means two, it basically means two coccus. So, some bacteria may be arranged in pairs, we call them as diplococci. And some may be arranged in groups, those are called as staphylococci or like a bunch of grapes. Some may form a tetrad or a cuboid, and sometimes some bacteria like streptococci may lie in long to short chains. That means streptococci, that means cocci in chains. So, all these names have been derived from how they are arranged. The other morphology that we can see is one is called as coccobacillus. Now, it is not a bacilli, not a cocci. Somewhere in between, it is called as coccobacilli. Now, if you look at this, slightly larger, slightly oblong shape, but it is not as long as bacilli. Hence, it is called as coccobacilli. If it is a long bacteria, we call it as bacilli or sometimes also called as rods because they look like rods. So, when you will be reading about various bacteria, sometimes you might come across the text where it says GNR or gram negative rods. It also means gram negative bacilli. Now, these bacilli can also be arranged in pairs called as diplobacilli or in stacks, which are called as palisades. Some bacteria may be enlarged, some may have a curved shape. A very common example is Vibrio or comma shaped, or it could appear like a corkscrew. Look at this picture, it appears like a corkscrew with a helical pattern. So, based on these types, we can classify the bacteria according to the shapes. Again, I have already said that based on gram stain, we can classify into either gram negative or gram positive bacilli. Now, this is a picture of a gram stain. I think you can observe there are some bacteria blue to purple in color and they are shaped as a How are they shaped? They are circular in small and circular, those are cocci. And you can see these bacteria which are pink to red in color and they are long rod like shape. These are called as. So, what can we see in this picture? You can see gram positive cocci and how it is the arrangement in clusters, and we can see gram negative bacilli. So, why I am telling you this? You need to be familiar with how the reports are being reported to the clinicians. This is how these are reported. Okay. Now, let us look at the structure of the bacteria. Now, bacterial structure, I think you may have studied a little bit in your plus 2 in biology. I think you can help me out over here. What are the structures the bacteria have? First is it's a unicellular, right? And it will contain a genetic material, that is the DNA. In bacteria, the DNA is a closed and circular pattern. It's not an open-ended. Additionally, there might be other genetic material which are called as plasmids. Those are called as extra chromosomal. So chromosomal DNA means that is the main genetic material. Other than that, any circular DNA if present, we call it as plasmid. Next, what does it contain in the cytoplasm? There is another organelle called as mesosomes. Now, what is the function of mesosomes? It acts like the mitochondria of an eukaryotic cell. That is, it produces the energy required for the bacterial metabolism. Other organelles may include a vacuole. What is the function of a vacuole? It engulfs any nutrient material for absorption and metabolism. The other important organelles would include ribosomes and RNA. So, what are the functions of ribosomes and RNA? They are involved in protein synthesis. Now, that encompasses the cytoplasm. Now, if we look at outside the cytoplasm, there is something called as the cell membrane. And outside the cell membrane, there is a thick layer which is called as cell wall. Other than that, outside the cell wall, you can see very thin hair-like structures. These are called as Pili or Fimbriae. And additionally, you can see long filamentous structure coming out. These are called as flagella. Now, let us look at all this one by one. Now, this is the ultrastructure of a cytoplasm. And what does it contains? It contains the DNA and RNA, polypeptides, ribosomes, mRNA, mesosomes, etc. Next comes is the cell wall. Now, this is the ultrastructure of the cell wall. It's a বলো এটা তো পড়েছো তো ফার্স্ট ইয়ারও তো পড়েছো সেল ওয়ালে কি থাকে লিপিডস and so the hydrophobic end would be outside and the hydrophilic end will be inside, and it will have what are these called as এটা কি এটিপি সিনথেস কি করে it produces the ATP and there are some transporter protein. What is the function of the transporter protein? It allows specific material to enter within the cell, and this is the main life source of the bacterial cell. Now, next comes the cell wall. Now, look at this picture. Can you appreciate any difference between these two types of bacteria? What is the difference? There is a blue color bacteria and there is a red color bacteria. This cell, what is the biggest difference? There is something bluish in color that is very huge in size, whereas over here the same material is present but the size is very small, right? And there is an additional layer on the outside. Can you appreciate the difference? Okay. Now, this is the main difference between a gram positive and gram negative cell wall. Let us look at more closely. This is a gram negative bacteria. If we take out this cell membrane and cell wall. We can see this is the cell membrane, and outside the cell membrane is a thick cell wall which is made up of what is a peptidoglycan? Peptidoglycan is a mesh structure made of two main important material. One is called as N-acetyl-muramic acid, N-acetyl glucosone. So they are arranged in a crisscross like pattern. Crisscross like a web or জাল দেখেছো তো স্ট্রেনার বাড়িতে চা ফা স্ট্রেন করেছো তো সেই স্ট্রেনারে যেমন প্যাটার্নটা থাকে সেরকম প্যাটার্ন থাকে উইথ আ ইন্টারলকিং প্যাটার্ন furthermore look at the size of the cell wall this peptidoglycan layer it's huge in case of gram positive about 10 to 25 nanometer. Furthermore, it will also have something called as teichoic acid. If it is present only in the cell wall, it is called as teichoic acid, and if the same is connected to the cell membrane with a lipid material, it is called as lipoteichoic acid. This teichoic acid enhances the virulence in case of gram positive bacteria. Now let us look at the gram negative bacteria. This is the cell membrane. This is the peptidoglycan layer. Now I think you can appreciate how small it is. And outside this peptidoglycan, we have what is called as the outer membrane layer. It contains the below which is the lipopolysaccharide layer. As the name suggests, lipopolysaccharide will have lipid, phosphate, and saccharides. And this lipopolysaccharide layer gives the virulence to the gram-negative bacteria. In gram-negative infections, there is something called as endotoxemia or toxin. So this acts as the toxin. Now, very briefly, I will make you understand why gram staining is important. In gram staining, have you performed gram staining? No. Okay. So in gram staining, what we do? First, we add a particular dye called as crystal violet. It enters into the cell of the bacteria. Okay. Then we use gram iodine, which acts like a mordant, and it stabilizes the crystal violet within the cell. After that, what we do is we use either alcohol or acetone to decolorize. When decolorization, what happens is this peptidoglycan layer is acted upon by acetone or alcohol and it breaks. So what will happen? Anything that is inside the cell will come out if this peptidoglycan layer breaks down. That is the main objective. And after that, we use another color. This is called as counter stain. Now, the timing of decoloration is very important. Let us suppose we use alcohol for a particular time. Let's say 2 to 3 seconds. What happens is, in case of gram-negative bacteria, the outer membrane is made up of lipids. So these are easily dissolved by the alcohol or acetone. Furthermore, the peptidoglycan layer breaks. And when it breaks down, what will happen? All the crystal violet iodine complexes that are present within the cell will come out of the bacteria, and the bacteria will lose all the primary stain. So when we use another stain or the counter stain, the bacteria will now take up the counter stain. Hence, the gram-positive bacteria will retain the primary stain, but the gram-negative bacteria will lose the primary stain and take up the secondary stain. The secondary stain is commonly used as safranin. Hence, the gram-negative bacteria appears as red to pink because safranin is red to pink in color. Have you understood? Now, this is very important. If I go on decolorizing with alcohol, what will happen? At one point of time, even the gram-positive peptidoglycan layer will also be broken. So then, even the gram-positive bacteria will also take up the counter stain and appear as gram-negative. Similarly, if I just eat in alcohol for a very few seconds or don't decolorize at all, what will happen? The gram-negative bacteria will also appear as gram-positive. Now, if you have understood this, I think you can now appreciate that the reports of gram stain will be dependent on the cell wall in both gram-positive and gram-negative bacteria. Why this is important medically? See, when a patient is infected, we don't wait for the reports. We start treatment with antibiotics, right? Most of the antibiotics will act on the cell wall. If the antibiotic is effective, what will happen to the bacterial cell wall? The bacterial cell wall will be damaged. Now, if I take the clinical sample from that patient and do a gram stain, will it appear normal or it will appear abnormal? Obviously, it will appear abnormal. Even gram positive bacteria may appear as gram variable or gram negative bacteria. So, this is very important. Okay. Now, some bacteria will have another covering outside the cell wall. This is called as capsule. Most of the capsules are polysaccharides. Saccharides means of carbohydrate origin. Now, what does this capsule do? It protects the bacteria from the environment. Not only it protects the bacteria from environment, inside the human body, it also protects the bacteria. What are the normal defense mechanisms? Have you heard about opsonization? What is opsonization? So basically, what is opsonization? পড়েছো তো? Opsonization and phagocytosis. Phagocytosis পড়েছো? Phagocytosis কি হয়? The engulfment হয় তো, engulfment এর আগে কি হয়? Opsonization. That means basically the bacteria is tagged and then phagocytosis occurs. This capsule prevents this opsonization. So if a bacteria is not opsonized, the chances are the phagocytosis will be less. If a bacteria is not phagocytosed, in the human body, which cells do phagocytosis? Which cells? Most commonly are polymorphonucleus white blood cells. তাই তো? They are involved in phagocytosis. The phagocytosis is one of the way by which the bacteria controls the human body controls the number of bacteria. So if a capsulated organism causes infection, the chances are the body's normal defense mechanism may not work. It will evade phagocytosis. Hence, the infection will spread very rapidly. In some bacteria, this capsule is not well formed. It is irregular in shape and it is called as slime layer. This slime layer, if many bacteria are there all having slime layer, they will form what is called as biofilm. Now, what is biofilm? A very common example is whenever there is a water leakage, have you seen the pipes have greenish accumulation of alga over here, pipes or any water source? So, those are the biofilms. So, this thick material is called as biofilm in which the bacteria can survive. The advantage of the biofilm is that it not only provides nutrition, it also protects the bacteria from environment and antibiotics. So, this is a natural phenomenon. But in medical terms, why this is important? During therapy, many times, many foreign bodies are placed inside the human body. It may be a urinary catheter, it may be a central line catheter for medication or monitoring, whatever it may be, we have to place. Or it could be even an endotracheal tube for the patients who have need for mechanical ventilation. What happens is these bacteria will colonize these foreign devices and form biofilm. And when this biofilm forms, what will happen? You can give any amount of antibiotics to the patient. That antibiotic will not be effective because the organisms have formed a biofilm. Okay. Another important appendage is called as flagella. Now, what is flagella? Flagella is what is the function of flagella? Yes. Now, why is it required? Why bacterial motility is required? Why bacterial motility is required? Acha, human beings, why human beings move from one place to other? In search of food, right? Even the bacteria do so. It will move from a nutrient deficient area to a nutrient rich area. How does it move? With the help of flagella. Now, what is a flagella? If we look at the ultrastructure, we have the filament which actually moves in a circular pattern so that the bacteria can move. The filament, and there is a basal body. This basal body is in the cell membrane itself. There is a mechanism by which energy is provided to the basal body, and this basal body moves the filament with the help of a hook. This is how a flagella works. Many bacteria will have very different types of flagella. Hence, another type of classification can also be done based on the flagella. Like, a bacteria might have a single flagella at one end. It is called as monotrichous. Mono means one, trichous means hair, because flagella looks like a hair. So, it is called as monotrichous. Or it may have a tuft, many flagella at one end. It is called as lopotrichous. It may have flagella at both ends. It is called as amphitrichous. Or the bacteria may have flagella all the way around. It is called as periflagella. Peri means surrounding. Okay. Other than flagella, if you remember, there are many thin hair-like structures all around the bacteria. These are called as Fimbriae or Pili. Now they have very specific function. The most important function is, any idea? It is called as adhesion. Okay. Let me give you an example. I think you have heard about Escherichia coli. This is a very common bacteria and it is very commonly implicated as a cause of urinary tract infections. Now, how does the urinary tract infection occurs? The bacteria enters through the urethra, moves through the urethra and enters into the bladder. And inside the bladder, it multiplies. And thus UTI happens, right? So, it has to move through the urethra. What normally happens is, when human beings urinate, the urine moves from bladder to urethra into the outside. So the bacteria has to move against the flow of urine. Agreed? So how does it move? With the help of a particular Fimbriae is called as a P. Fimbriae. This Fimbriae attaches the bacteria to the urothelium, that is the urethral endothelium. It attaches the bacteria. So even if the urine passes over, the bacteria is not dislodged. Slowly and steadily it moves upstream and causes urinary tract infection. So if a E. coli has P. fimbriae, it can cause urinary tract infection. And if a particular E. coli does not have P. fimbriae, can it cause UTI? Normally, no. If it gets implanted into the bladder by any other means, it can cause. But the natural route, it cannot cause the UTI. Okay. So that is a very important function of Fimbriae. Another type of Fimbriae or Pili is called as sex Pili. Why is it called sex Pili? Sometimes two bacteria may come in close contact with each other, and this sex Pili forms a bridge between the two bacilli. What kind of genetic material? Recall, I have already told you there is something called as extra chromosomal DNA called as plasmids. These plasmids can be transferred from one bacteria to the other. This is one of the very common mechanisms by which antibiotic resistance genes are transferred from one bacteria to the other. You will be taught it in much depth in later classes. Morphologically, we can observe Fimbriae by forming in some bacteria. If we culture them on liquid media, we can see them forming surface pellicle. This surface pellicle is because of the Fimbriae. Okay. Another very important aspect is called as bacterial spores. Now, what is a bacterial spore? What is a spore? Any idea? Have you heard about hibernation? Obviously, some animals hibernate. When does it hibernate? When the environment gets too difficult to survive. So, what are the preparations like a bear? It hibernates. What preparation does it take before hibernating? It will hunt and eat a lot of food to survive the period of hibernation, and it will enter into a cave or a mountain cave to overcome the winters, right? A similar phenomenon is also seen with bacteria. Whenever there is a environmental change, it could be nutrient deficiency, it could be harsh temperature, it could be toxic chemicals. If the bacteria is exposed to all these things, what it will do? It will try to protect itself. The normal mechanism stops and the bacteria condenses its genetic material into forming what is called as the core. The core has the DNA, it survives. And to protect the core, there is a formation of cortex. The polypeptides, polysaccharides, all gets condensed to form a cortex. This cortex is very hard, hence, it protects the inner core of genetic material. There are other formations like the coat, outer membrane, and exosporium, all adding to the further integrity of the spore structure. So, there are two forms of the bacteria. One is called as the vegetative bacteria. What, what is the vegetative bacteria? All the morphology I told you about, those are the parts of the vegetative bacteria. And if any harsh environment is exposed to that vegetative bacteria, it will form spore. These are the hardened spores and it will resist. Again, based on the spore, we can classify many bacteria because in some bacteria, the spores may be bulging. That is, you look at this vegetative skeleton. This is not the bacteria anymore. It is only the skeleton of the bacteria and the spore bulges out of the skeleton. This is called as bulging spore. Or it might be small enough so as not to bulge. It is called as non-bulged. And based on the position, it could be terminal, that is at the end of the bacilli, or it could be central, right in the center, or subterminal, that is somewhere between end and the center. Based on this, obviously, again another classification is there, like non-bulging, oval, terminal. Okay, the shape may be oval or round in case of various bacteria. Two phenomena occurs. One is called as sporulation. When a vegetative bacteria turns into spore, we call it as sporulation. And the reverse, that is, when the spore again becomes a vegetative bacteria, we call it as germination. Now, having understood this, we also need to understand bacterial division. The bacteria reproduces by binary division. What is binary division? One cell multiplies into two cells. So, I think you can see in this animation how one cell divides into two cells. And if we look at the bacterial, this is called as the bacterial growth curve. Now, what is bacterial growth curve? If we take a single bacteria in a culture media which contains nutrients and all other essential things, and we incubate them at a particular temperature, what will happen? That single bacteria will multiply into two. Then those two bacteria will multiply into four, and this will go on. How long? Initially, when the bacteria gets the nutrients, it will start to prepare for multiplication. The numbers don't increase, but they start to prepare. This is called as the lag phase. If we count the number of bacteria, you can see the number is constant. Say we have taken 100 bacteria. In this lag phase, the number of bacteria will remain 100. But what these bacteria are doing, it is taking up the nutrients and it is preparing for cell division. Thereafter, we can see there is an exponential growth. This is called as the log phase. Remember, logarithmic scales in mathematics is used to denote exponential. So, this is the exponential growth where those 100 bacteria will now become maybe 1000 or 10,000. Thereafter, what happens? The nutrient becomes depleted, or the amount of nutrients in the media is taken up. So, now the nutrients will be more limited. So, what will happen to the cell division? It will come down, it will not stop, it will come down. So, we get a stationary phase. Say about 10,000 bacteria have from 100 to 10,000, now it will remain at 10,000. Some multiplication will be there and some bacteria will start dying. So, overall number remains constant. Thereafter, we come to the death phase. What happens in this death phase? The nutrient is almost depleted, and when these nutrients are depleted, what will happen to the bacterial cell? They will die because nutrients are not there. When the bacteria dies, it will release enzymes, exotoxins, and the whole environment will be more harsh. More and more bacteria will start dying. This is called as the death phase or declining phase. Have you understood? Okay. Now, two more important terminology that you must understand. One is called as total count, one is called as viable count. What is total count? Suppose this is a bottle in a culture media. Initially, I have taken 100 bacteria. So, in the lag phase, there will be 100. Say in the log phase, it has multiplied into 10,000. Then the number will start going down. Okay. But at each point, if I take a particular sample from the liquid media and count the number of bacteria, we will get what is called as total count. Both living, both dead. But if we reculture them, we can get what is called as the viable count, that is number of living bacteria. So, if we apply this, what will happen? Over here, both total count and viable count will be same. In the stationary phase, both total count and viable count will also be same. Both will be multiplying. But in the stationary phase, the viable count will be in a plateau. That is, the number of viable bacteria will remain stable. But the total count will keep on increasing. Why? Because some of the bacteria are dying. And in the death phase, viable bacteria will go down. However, total count will remain constant. Understood? Okay. Now, if we culture these bacteria artificially or in vitro, their number will increase, right? Look at this picture on an agar plate. If I put a single bacterial cell wall, after about 12 hours, the number increases to almost about 17,000. Why is this time important? Because there is something called as generation time. That is the time required for one bacterium to multiply into two bacteria. Every bacteria has a different generation time. E. coli has a generation time of 20 minutes. Mycobacterium tuberculosis, which causes tuberculosis, has a generation time about 24 hours. So, depending on the generation time, the period of incubation in the laboratory depends. Furthermore, as I have already said, various environmental factors matters in case of bacterial growth. Let us look at those factors. First is oxygen. Now, why oxygen is important? Why oxygen is important? অক্সিজেন কি করে? পড়েছো তো, ফিজিওলজি বায়োকেমিস্ট্রি তো পড়েছো তো। লাস্ট ইলেকট্রন অ্যাকসেপ্টর কে? এটিপি সিনথেসিসে। So, it is required for ATP synthesis in case of aerobic metabolism. So, some bacteria are called as obligate aerobe. Their metabolism is by aerobic method. Some bacteria are called as anaerobe. They do not undergo any aerobic metabolism. They only have fermentative metabolism, that is anaerobic metabolism, right? There are some bacteria which survive in aerobic conditions and can also tolerate anaerobic. Most of the human bacteria causing human infections are these type of bacteria. They are called as facultative anaerobe. So based on this, we can divide the bacteria into obligate aerobes, obligate anaerobes, and facultative anaerobes. Okay. Next comes carbon dioxide. Some bacteria are, that is, they love carbon dioxide. Hence, they are called as capnophilic. They require a higher percentage of carbon dioxide for their growth. And some bacteria, they require very small amount of oxygen, not the normal, say 20-21% of oxygen in the air. They are called as microaerophilic. Microaerophilic. They need oxygen but in a very small amount. What will be the temperature requirement? In environment, we can find bacteria growing at any temperature. Below 10 degrees centigrade, more than 90 degrees centigrade. But all the bacteria which cause human infection will grow at temperature round about human body temperature. That is 25 to 40 degrees centigrade. This is the temperature range where medically important bacteria grow. Next comes pH. Most of the bacteria will grow at which pH? In the environment, you can find bacteria growing in acidic environment, in alkaline environment. But what will happen in case of medically important bacteria? They will grow at a neutral pH because human body has a neutral pH. However, you must understand some bacteria will have the ability to tolerate different pHs. For example, if a bacteria has to cause intestinal infection, it has got to pass through the gastric acid. Gastric acid has a, because it's an acid, it will have an acidic pH. And if you go to the duodenum, we have a alkaline pH. So any bacteria which can cause intestinal infection will have to tolerate both acidic and alkaline pH. Next comes light. Light changes various aspects. For example, some of the microbacteria will give color when exposed to light, may not give a color. Or next comes osmotic effect. What is osmotic effect? What is osmosis? What happens to a cell when we expose it to a, say, hyperosmolar solution? It will shrink. And what will happen if we expose to a hypoosmolar solution? It will swell up and it will ultimately, the cell membrane will rupture. Same thing happens with the bacteria. If we put the bacteria in a hypertonic saline, that is a saline concentration more than required, what will happen? It will shrink. And if we put it in a hypotonic solution, what will happen? It will swell up. This begs the question, medically important bacteria, what will be the normal osmolarity? What is the normal osmolarity? What is the osmolarity in blood? So, in human beings, what do we give? We give normal saline, right? So, all medical bacteria will be able to tolerate physiological saline. That is the importance. তাহলে কোনো ক্লিনিক্যাল স্যাম্পল যদি পাঠাতে হয় কালচারের জন্য, if you put hypertonic saline, what will happen? The bacteria will die, culture negative আসবে। তাহলে whenever we have to use physiological or normal saline. This is the main takeaway point. Okay. Other things that can disrupt is mechanical stress, like ultrasound, ultrasonication can damage the bacteria, or moisture and desiccation. Moisture, that is water, is required for the growth of bacteria, and if we maintain a dry environment, most of the bacteria will die. Now, these points are important, especially in the hospital setting when we are trying to prevent infections. This is why, if you go to the hospital, you will see every day the floors are wiped and then cleaned till dry. And it is ensured that none of the surfaces remain wet, because if it remains wet, environmental bacteria will grow. Okay. So, this is the picture which shows how metabolism occurs. On the right hand side, you can see aerobic respiration, and on the left hand side, you can see anaerobic or fermentative metabolism. So, aerobic bacteria will have aerobic respiration, anaerobic will have fermentative respiration. Facultative anaerobes will have both. Finally, I have already told you, when we classify medically important bacteria phenetically, we also try to understand the various metabolic or the metabolism of the bacteria. This is a test. You can, if you can see, the top one is an uninoculated tube and the bottom one is an inoculated tube, and you can see some of the changes of the color of the media. Now, why these changes occur? Because of the various metabolism, like whether the glucose is utilized or not, whether specific carbohydrates are utilized or not, or whether urease is produced or not. If the urease is produced, what will it do? It will degrade urea, or whether citrate is utilized or not. These are specific metabolic pathways which we can test to identify specific groups of bacteria. Now, finally, this is the chart which shows how the cell wall or the peptidoglycan of the bacteria is produced by interlinking of the DLR to producing N-acetylmuramic acid and N-acetylglucosamine, and these are interlinked with the PEP chain. Now, this metabolism is very important, because when you will be learning about the antibiotics, you will learn that many of the antibiotics inhibit this peptidoglycan synthesis. And the final aspect of the morphology is this. What is this? Anybody? Any idea? This is the electron microscopic picture of the bacteria. Good. So, what are bacteriophages? These are viruses which prey on bacteria. So, if you think that only bacteria can survive in the environment without any predation, it is not true. Viruses prey on the bacteria, and they prey on the cell wall of the bacteria. These things are also utilized in many research technologies that you will be acquainted later on. So, what you need to understand, there are particular viruses which prey on bacteria. These are called as bacteriophages. And finally, this is a very small list of the various bacteria which cause specific infections based on the specific syndrome, like meningitis, otitis media. Otitis means ear, and media means internal ear. Pneumonia, that is the infection of the lung. Skin infections, sexually transmitted infections, urinary tract infections, intestinal infections, and sinusitis. This is not a full list, this is just a general list. Okay. To recapitulate, the important point that you must remember is, morphologically we can classify bacteria in various methods, but from the diagnostic point of view, morphological classification is very important for early identification so that treatment can be initiated. Okay.